Hi, everyone. I'm Brent Allman. I'm a postdoc at UT Austin within host dynamics, within host evolution of viruses for me primarily.
I'm Ashley Love from the University of Connecticut in Sarah Canuti's lab, and I'm interested in why hosts vary and how they respond to infection, looking at factors like diet and also social cues from the environment.
Hi. Oh, hello. My name is Allison. I'm a postdoc over at Vanderbilt, Nan Tate's lab, studying molecular dynamics during infection and host pathogen interactions.
Hi, I'm Dami Peck, and I'm a postdoc with Megan Gresher at Cornell University, and I'm interested in why malaria parasites can't proliferate faster. So I guess anyone have like any questions to start off the thing, or should we make something up?
Well, first, for questions, we would like to start, if you're trainee, grad student, or maybe this is your first EEID, we invite you to speak first, and then we can kind of dive into discussions with everybody. So does anyone have what do they think is kind of an important burning question in like within host dynamics?
Yeah, we'll wait for the more senior people.
So should we just start?
Oh.
Hi, so I guess this is kind of a question for Daniel Metz. So that's fascinating work on the demographics of trematodes and specifically thinking about how, this life stage within the snail host is influenced depending on mortality and birth rates and longevity. I'm curious if you are trying to do this modeling with the life stage that's within the bird host, and if you have any predictions on how the effective population size of adult worms or adult trematodes within the birds might be influencing some of those dynamics that we're seeing for those taxa.
Should I just yell? Cool. Yeah. So one of the challenges of using that kind of a model structure for the adult population is that we don't have any expectation that it's stage structured in the final host. We've got a lot of like immigration, probably constant immigration from diet, if they're trophically transmitted, something like that, death rates that can be long-lived, and there's no auto recruitment going on. So they're not building up their population by their own efforts. It's a little bit incidental when they're coming in for these trematodes that are living in the guts or blood flukes or things like that. So there's no expectation that, from my perspective, at least, that a model like a stage structured infrademographic model like I was presenting would be particularly useful for like a typical parasite like that. But then as far as does what's happening in the bird host influence the overall demography of the first intermediate host? Absolutely. Probably doesn't much influence what's going on in a particular snail though, since what you have with one snail is you have one clone. It's like a single worm infects that snail, and then that worm clones itself over and over until it just fills up the available habitat. And then that process of that continual auto recruitment is what kind of allows this stage structure to arise. It's kind of like a closed population, which is why that simple demographic model works really well. Thank you. That's a cool question.
Are there any more trainees that have questions either for the speakers or maybe for the room in general about questions within host dynamics broadly before we get to Matt?
Yes.
Thanks, Ta.
Thank you, Ta.
Yeah, so I was just thinking about, so this session we're talking about within host, there's a lot of variability, and the morning session with Alex talk about multi-scale, and so I'm just wondering what could be a good framework for us to sort of bridging the gap between within host versus population level way of thinking and studying diseases.
Yeah, does anyone have any thoughts on that? Kind of how we're bridging kind of this like within host versus across host? Yeah. Because yeah, obviously it's very complicated, right? That's our answer to everything also in ecology too. It depends and it's complicated. Yeah.
Yeah, I think, I'm sorry for my voice. I think that's the right answer. I've tried to do this, linking within hosts to say population scale patterns, and it's really hard because different things seem to matter at different scales. Like, it just, the interactions between parasites are different, the host, like interactions are, have different sort of levels of importance. The external environment is really important for population scale dynamics, less important for within host dynamics. So I don't have a good answer besides the fact that it's not as easy as it seems, but that it's really, I think it's a really good question that we have to keep continuing to figure out how to link those two things, because obviously they affect each other. But I'm not sure if I have a good question, but it depends. And you have to really sort of find good data sets that have information at these multiple scales as well. And experiments really help, because it's really hard to observe these really sort of intricate interactions in observational data.
All right, I'd like to encourage folks to consider it from both directions in terms of linkages, both into and out of the within host level. I think at least from my natural inclination, is more to focus on the consequences of within host dynamics for between host transmission. But I think it's also important to remember that within those dynamics can be also shaped by the epidemiological context, what's going on in the population, the host population level seeds what goes on in the within host level. There can also be important effects in that direction as well. So I'll put a plug in for the field to consider both the inputs and outputs of the within host level.
I wanted to respond to, I think it was Evie over there. So your point about, well, it depends, which is always true, of course, but I think there have been a lot of really important developments in community ecology on coexistence theory in the last 20 years or so that can help us understand not just within host coexistence, but also coexistence within populations and within hosts, things like scale transition theory and some aspects of meta community theory and so on. And these theories can help to at least identify the key parameters that matter for coexistence outcomes and help us to design those experiments. Like, we need to be measuring within versus between species competition coefficients and things like that, as opposed to measuring just the outcome of competition.
Hi, I think this fantastic suite of talks in this within host session really illustrated some further developments in the last 20 years, sort of following on from what Aaron started about reflections on the last 20 years, just as we did for this morning's session. First and foremost, the last 20 years, we really now can sort of begin to characterize those within host, the symbiont communities, both endogenous and exogenous, to use Yasmin's terminologies, like revolutionary, right? In terms of understanding what we previously thought was a mammal phenotype being this holobiont, right? That's been huge in the last 20 years, and these talks really got at that. But equally, this idea of immune regulation being as important as, getting a potent immune response going is really important for surviving infection, right? But regulating the immune system so you don't kill yourself with it. We've long known that that was important, but I feel like, for example, in Avery's talk, showing that it's at the level of the single cell, it's got the danger, but it also is self-regulating. That kind of insight has also emerged in the last 20 years. So I just wanted to note the sort of the long view on what we've heard this afternoon, really reflecting incredible progress. It maybe adds to the complexity, right? If you have to think about that whole bajillion of community members that are symbiotic within every host who's fighting the infection you want to think about, right? That's a layer of complexity that might make you want to cry. But also the immune system being self-regulatory at a bunch of different levels is really important and exciting. So I maybe more challenges for us, but I'm super excited about those aspects that we've heard about this afternoon.
Yeah, any other reflections maybe on the past 20 years? Yeah.
I don't think I can talk about the last 20 years. But just to sort of continue what Andrea was saying, there is we've learned a lot about within host dynamics, particularly in the vertebrates system. And coming from a vector-borne disease perspective, I think there's a lot more to be gained from what's happening within the invertebrate hosts, right? So they also have their microbiota, they also have their immune system. And I think there's just a lot of things that we don't know from a biological perspective, but then, you know, obviously we need the models to help really understand that part of the transmission cycle.
Along those same lines, I saw both sides of the microbiome within hosts that really fascinate me, where we're looking at how the microbiome is perhaps regulating the immune system of the host. But then we also saw that the microbiome may be directly competing or inhibiting pathogen colonization. I think these direct and indirect mechanisms of the microbiome are really important to investigate the different ladders of how they could be influencing within infection.
Thanks.
Yeah, so I really enjoyed the talks today, and I felt like one thing that kind of emerged is that that kind of called upon some of the stuff that came up this morning was thinking about how the immune system alters these trade-offs between host mortality and transmission. And I think a lot of the talks addressed portions of that, and I think it's really hard to do in the same system. Maybe the mycoplasma started to get at that. And I think one question that sticks in my mind, possibly related, possibly not, is thinking about this tuning of the immune system and whether there are any generalities there, or whether there's so much idiosyncrasy across host backgrounds or microbiome or other environmental insults. Like, are there any general principles we can draw from that, or is it all highly variable? Thanks.
So sorry if this sounds self-serving, because thank you for the intro with the mycoplasma system. But I think, to me, at the end of the day, it's transmission experiments. And that's what really matters to me. And I think that would be so fun to see more transmission experiments to see how these dynamics within a host actually translate to the transmission.
When you say transmission experiments, you mean manipulating different aspects of a singular host infection to see how that influences onward transmission, or were there other aspects that? Okay. Does anyone have any thoughts on that line in particular?
I think you got the mic first. I just wanted to add my support for that, for more of these experiments. I think one of the challenges from my perspective is that infectiousness has no natural units. And so, we have to measure it by a combination of within host dynamics, behaviors, contacts, and actually look at the probability of a transmission occurring. So anyway, just a lot of support for that kind of idea. I think creativity there, decreases in cost, increases in scale, and consideration of ethics when doing so is going to open up a lot of new questions and answers for us.
Yeah, I would also echo the call to, have more and more thoughtful transmission experiments done, but equally important, making sure that we analyze them as thoughtfully as possible to get the maximum value out of those data, because of the ethical considerations. More broadly, like to the point of the complexity of connecting across scales, obviously this comes down to, what governs fitness at the between host scale, and how does that connect to fitness at the within host scale. And so whatever we can do to understand the traits on the pathogen side and the host side that govern the successful transmission event will help us to sort of navigate that complexity. On that note, I would just say a lot of this comes down to pathogen life history. And some pathogens basically interact with the host in a relatively simple way at all the action happens in your throat, say, or, somewhere near the surface in the bloodstream. Others have a much more complex life history that interacts with multiple host compartments, and we may not be getting our most convenient measurements of the within host dynamics in the parts that matter for the onward transmission. And so sort of mindfulness of the possible sort of compartmental structure, the fact that a host is not a bag of pathogens, and ideally harnessing both experimental, empirical, and analytic tools to get insight into that might help to, bridge these two scales.
So I guess I'll double down on that in the sense of, now that I work on opportunistic bacterial pathogens, I'm not sure what a compartment means. If a lineage is leaving the human body, successfully having been a pathogen, and it's going to live in a pasture and soil, and then maybe in water, and then come back. I don't know what that even means. How am I going to measure that? What is it transmitting between, and what can I actually, model to have that be relevant? So I'm very interested in tracing a lineage and how successful it is, which is damn hard, right? Because I'm kind of looking at evolvability, which is a bit of a trigger word. Most people don't even believe it exists. So I think this is pretty challenging when you talk about things that are not going directly from host to host, but they're going to live in some other environment for sometimes very many generations and then coming back. Is that still the same creature, or is that, what am I looking at that point?
Yeah, go ahead.
So I just want to ask my question. I just gonna ask a question. This is not an announcement. But I wanted to return back to some of the thinking about the immune system, and this is actually a question that I asked Anne last night that I think Andrea alluded to, and I think came up in Avery's talk. And I think one of the things that's been really exciting about what this field has done has been the recognition of dynamical thinking, right? Not just the events, the interventions, but the progression of those things, the dynamics of populations, what that means to predictability, to transmission, to the likelihood of interruption and prevention of disease. But I'm wondering, given the sort of watershed moment that we've had over the last several years, the general public has become increasingly aware of these things. There's one of the things that was on the front, the front page was the role of immune pathology and immunodynamics in the course of disease and the severity of disease and the duration of disease, right? In viral loads. And so I'm wondering if you guys see this recognition as sort of am I just noticing and you guys have known this whole time? Or is it, is there sort of a watershed moment where this is a time to really focus on, the dynamics of immune systems and actually kind of bring that dynamical thinking, these ideas of multiple stable states, the predictability and forecasting of disease outcomes in individuals? That's the question I want to put to this crowd, especially to the experts that we have in the room, because I rarely get to have you guys all here all at once.
Let Anne speak.
Yeah, I think one of the things that I'm starting to really appreciate is not just, which cytokines get produced, but when and how often. Like, for example, in SARS-CoV-2, right? Everybody produces type 1 interferons, but if it's delayed in the production, then you get this really crazy pathology. Whereas if you produce it really quickly, then, you recover really quickly. And we saw with Avery August's talk about, the strength of T cell receptor signaling, right? You get these combinatorics of transcription factors depending on the strength that's signaling. That's ultimately going to basically incredibly influence both the polarization of responses, but also the resolution of those responses, right? And what the fundamental profiles are. And so I think this all comes down to kind of these really fine scale dynamics where small changes in early events can lead to these really discrete infection outcomes. And I think that's something, somebody who models a lot of within host dynamics, that's something we think about a lot. But I think even, general immunology is really starting to appreciate those things as well.
Yeah, if I could, can I? Yeah. Well, I could build off of Anne's point there. I think there's something that I have realized, and I think other people have started to realize, is that actually like the theoretical immunology literature, which is like 20 years old, it was aware of many of these things a long time ago. And as a field, one of the things I think we're starting to do is to look at that literature a little bit more, and to broaden. I think we started with really influential work that was analogizing within host dynamics to things that as ecologists we understood well, with sort of predator prey, and now our analogies are getting richer as we sort of draw on the theoretical immunology literature. And so I think one really thing I think is happening and we'll be excited to see going forward is doing more of this integration where we take very seriously those theoretical models that exist, that were developed by immunologists, and bringing them into, like, we build great epidemiological models and they build great immunological models, and getting those to talk to each other to ask what are the consequences of something like bistability, multiple stable states in immune dynamics. What are the implications of those for transmission, for virulence evolution? I think will be something that will be really interesting going forward.
Being here the longest and being one of the oldest, it's almost 4 o'clock, so I managed to squeeze in at the end. But my request will be, it's time to move away from the mean. It's time to embrace the variance. And I'm answering Matt's question here. Embrace the variation and why it is that some people respond like this and some people respond like that. And I really liked that last talk because of the complexities, but the results in the individual variation that we see. So please embrace that variance.
Thank you.
If I can just follow up on that, Matt, I think we're already starting to see some of the impacts of embracing that variance in fields that aren't centering our infectious disease dynamics. So something that I've been thinking about that follows our developments in understanding contextual immunology and outcomes of infections is that I've been working for a long time on the impacts of neonicotinoids in non-target insects. That's the world's most used insecticide, and at the time, the world's largest economy banned it based on quite shaky evidence. And what's emerged over time is that the main impact of those chemicals is that they're an immunosuppressant, and so you only see a major impact of them based on the presence or absence of certain pathogens. And that as our understanding of that immunological underpinning of outcome of infection grows, we're able to circle back to these other fields elsewhere in ecology, elsewhere in evolution, and actually help them understand these very real political challenges to society. So I think it's encouraging to hear how much support there is for that, seeing how it then spills over, for lack of a better term, into other aspects of our work.
Excellent. So first of all, I want to thank everybody for a fantastic day. This has been a busy day. It's been a big day. There's been a lot of science. But now I want to encourage everybody to go and embrace the variability that's up in the poster session. I want to thank our facilitators for a fantastic discussion in the afternoon.